right, looks like we've got a lot of people joining. Well, welcome everyone. I'm Jen Farmer, CEO of the Friedreich Ataxia Research Alliance, and FARA is excited to host our third community webinar this year. And I've got a great group of people joining me today for this community webinar. We have Barbara Tate, FARA's Chief Scientific Officer. We have Kellen Madden, FARA's Patient Engagement Manager. We have Chandra Trantham, who is a FARA ambassador and a PhD candidate at the University of Florida. And we have Becca Lang, who is an FA community member, mother to Arlie, Annie, and Abel. And Arlie and Abel both have FA. So thank you everyone for joining today. We have these, webinars so that we can have more of a conversational or interactive discussion around topics that are important to our community. And today we're focused on genetic therapies. And so Kellen is going to take us through an overview of what genetic therapies are. And we'll pause for some questions from Chandra and Becca. And then I will give an overview of how genetic therapies are being developed for FA. And again, we'll pause for some questions. And then Barbara will specifically cover what FARA is doing to support development of genetic therapies. And we'll take questions again at the end. For those of you joining online, there is a Q&A box. It should be at the bottom of your screen in the Zoom link. Encourage you to submit questions as we go. And again, we'll try and get to them. We also appreciate those of you who submitted questions in advance, and we will try and cover some of them in our Q&A as we go as well. So hopefully most of you are familiar with our Friedreich's ataxia drug development pipeline, which I'm showing here. In our previous webinar back in May, we focused on programs at the top of the drug development pipeline. And these treatment approaches are more traditional type drugs. They're small, most of them are small molecules. There's one protein replacement, but these are more traditional therapies. Today, we're focusing on the bottom half of the drug development pipeline on genetic therapies that are in development. And if you're familiar with this pipeline, you know that things that are over here on the left-hand side are earlier in development, and things over here on the right-hand side are closer to being available for as clinical therapies or approved medicines. A few important points about why we're having a special webinar just on genetic therapies. There are some important differences between small molecule type drugs and genetic therapies. One of the biggest ones is that small molecules, if you think of them as even like Advil or Tylenol that you might take for a headache, when you take them, they're oftentimes oral medicines or injectables even, but after a while they leave your system, right? They're washed out. Genetic therapies, as you'll learn from Kellen, are making a permanent change inside your cell. And so when genetic therapies are administered, they're non-reversible and oftentimes only given a single time. So this obviously has all kinds of implications as to the long and short-term effects of these medicines, but it also influences even the clinical trial designs. And what we've seen in clinical trials from small molecules are going to be different than what we see in clinical trials for genetic therapies. One example is the duration of the trial. Many times when we start with phase 1, phase 2 trials, they're short periods of time, a few days, a few weeks, maybe a few months. But in the case of genetic therapies, even a phase 1 early stage trial will take place over the course of many years. And it's actually regulated that genetic therapy trials be up to 5 years at present. The other thing to consider because of this non-reversible aspect of these therapies is that it may impact your ability to participate in future trials. With small molecules, again, they wash out, and typically there's a period of time that people have to wait when going from one trial to the next. But in the case of a genetic therapy trial, it may be that if you do a genetic therapy trial, you might not be able to do another one, or you might not be able to even take an approved product of a genetic therapy if you've been previously exposed to a genetic therapy in a clinical trial. And so there's some important distinctions that that make genetic therapies really special. We're going to talk about them today, but just as a background as to why we've carved out genetic therapies in our pipeline for today's conversation. So with that, I'm going to turn it over to Kellen, and Kellen is going to start at the beginning and really help give an overview as to, what we mean when we say genetic therapies. Kellen?
Thanks, Jen. So before we talk about genetic therapies, first we have to talk about genes and what genes are. We can go on to the next slide. So our bodies are made up of trillions of cells, and inside of each cell is a nucleus that contains DNA, and DNA is organized into units called genes. Genes contain the instructions for proteins, which are molecules that can help to build different elements of cells and tissues and organs. They help cells and tissues function, and they also help to determine some of our physical traits like height or eye color. To make protein, DNA is first transcribed into an intermediate molecule called RNA, and that's translated then into protein. And the protein is also involved in helping the cell function and helping to build the cell and determining physical traits. When a gene is mutated, this can cause issues in making RNA and making protein, or in both. In the case of FA, the gene that's mutated is the FXN gene, and the mutation is an extra repetition of the DNA letters GAA. Everyone's frataxin genes all have GAA repetitions, but in FA, they are repeated too many times, and this repetition prevents the RNA from being made from the DNA. And when not enough RNA is made, not enough frataxin protein is made, and that is what leads to the symptoms of FA. But since we know the genetic cause of FA, that means that there's potential to address FA at the DNA level. And there are two different ways that this could be done. The first is gene replacement, and the second is gene editing. There are two ways that these two types of genetic therapies can be delivered to the body, and that is ex vivo and in vivo. And we'll talk about all of these categories in the next few slides. So first up is gene replacement. In gene replacement, a healthy copy of the FXN gene is delivered to cells, and you can see that the original mutated gene remains present in the cells, and it's still not making enough RNA or enough protein. But now that healthy gene that's been delivered is also in the cell, and it's compensating for the native mutated genes by making its own frataxin protein. And you can see at the bottom of the slide, there's this little Amazon delivery truck that is delivering the gene to the cell. We'll talk about this in a couple more slides, but every type of genetic medicine needs some type of delivery vehicle. And in scientific terms, this is something that's called a vector. On the next slide, we'll talk about gene editing. And gene editing, similarly, you need to use a delivery vehicle or a vector, but in this case, you are delivering gene editing tools, which there are a lot of different types of molecular gene editing tools that are being researched. Some of them can cut DNA. So in the case of FA, it might be possible to cut out the extra GAA repeats of the native gene that is in the cells. So it's your own DNA in the case of gene editing that's being changed. And the theory is that once the extra GAA repeats are removed, that would allow the DNA to then make RNA, which would then make more frataxin protein. So on the previous slides, we just had an image of one cell and a little delivery vehicle delivering the genetic therapies to the cell, but it's a lot more complicated than that, as you can imagine. Again, we have 37 trillion cells in our body, and there are millions of cells within each organ. So there are millions of cells in the heart. There are millions of cells in the nervous system. And in terms of genetic therapies for FA, not only do we need to make sure that the genetic therapies get to the organs like the heart and the nervous system that we want to get them to, but we also have to make sure we get enough genetic therapy to enough of the cells, because it's not always going to be a one-to-one ratio where every cell definitely receives the genetic therapy. So this is why the delivery vehicles, or the vectors, are so important. And you may have heard in terms of gene therapy about adeno-associated viruses. So right now, viral vectors are the most commonly used vector to deliver genetic therapies into cells. Viruses are naturally very good at getting into cells. That's how viral infections happen. But in terms of genetic medicine, there isn't any viral DNA or viral genes in the virus. It's just the coat of the virus that's being used as the delivery truck to get to the genetic therapy, whether that's the healthy gene that's being used in gene replacement or the gene editing tools into the cells. Viruses create an issue that Jen kind of already brought up, where because it's a virus, you'll mount an immune response to the virus if you receive a viral-based gene therapy. And that means you might not be able to receive another viral-based gene therapy because you have antibodies that will neutralize it as soon as you see that same virus again. So that's why there's a lot of research going on for different types of vectors, including lipid nanoparticles and something called extracellular vesicles. And there's also a lot of research being done on the viral vectors to make sure that they're as safe and efficient as possible. So that was information about how we get the genetic tools or the genetic therapies into the different cells, but how do we actually get the genetic therapies into the body? There are two main ways that this happens. The first is something called in vivo, which means inside the body. And in this instance, the genetic therapy is directly injected into someone's body. The second is ex vivo, which means outside the body. And in this approach, cells are taken from someone's body, they're processed in a lab, and through that process, the genetic therapy, whether it's gene editing tools or the healthy gene for gene replacement, are added to the cells. The cells are allowed to grow a little bit more, and now all of those cells contain the genetic therapy, and they're put back into the body containing the genetic therapy. And on the next slide, you can see in terms of in vivo administration, so when the genetic therapy is added directly to the body, there are a couple different ways that that can be accomplished. One is intravenous or IV infusion, and in this type of administration, typically the genetic therapy would likely be able to reach tissues like the heart and the muscles. But it's thought, based on research that we have right now, that an intravenous infusion would not reach the nervous system. So it's also possible to directly inject a genetic therapy into the spinal fluid, which would be called an intrathecal injection, or into the brain in FA, likely the cerebellum, because that is the part of the brain most affected in FA, and this is called an intraparenchymal injection. And it's also possible to inject genetic therapy directly into the eyes as well. So this is, again, that summary slide that shows the different types of genetic therapy and the different way that it can be administered. There's gene replacement, which is adding the healthy gene into the cells. There's gene editing, which is editing someone's native mutated genes to somehow ameliorate the effect of the mutation. And then there's ex vivo, which is when you take the cells out of the body, treat them, and put them back in vivo, which is when you directly inject the genetic medicine into someone's body. And we have here examples of approved genetic therapies for several different rare diseases. It's really exciting that there have been approvals for other diseases in terms of genetic therapies, but we're still at a pretty early stage. So you can see there's Not too many examples on the screen, and most of them are gene replacement, adding in the healthy copy of the gene into a patient's cells. There's only one example of genetic gene editing that has been approved. And something that makes these examples a little bit distinct from FA is the fact that a lot of these diseases, there's only one tissue type that needs to be targeted or treated. So sickle cell disease, for example, that's a blood cell, that the blood cells are the cells in that disease that are affected, and that's just the only cell type that needs to be targeted in that disease. But for Friedreich's ataxia, we need to target the nervous system and the heart and potentially other parts of the body as well. All right, I think we can take some questions now from one of our moderators.
So if I got gene replacement therapy or gene editing and I later had children, would those edits or changes be passed down to my children?
So likely the edits or the changes that happen from genetic therapies would not be passed down to someone's children. As we just talked about, we have to be really specific when designing genetic therapies to make sure they go to the organ systems that we want them to go to. So most genetic therapies are not going to reach cells like egg cells or sperm cells that are what pass down people's DNA to their children.
Thank you. And then also, I know that you said that for genetic therapies, there needs to be a delivery vehicle to get it into the body where it needs to go. Is there like a delivery vehicle that can get to all the relevant cells in FA?
Right now, we don't think that there is, so there's a lot of research going on to address that issue. With the viral vectors, the AAV vectors, some of the vectors are really good at getting to the heart, for example. There aren't many vectors that we know that can cross the blood-brain barrier in terms of viral vectors, so that's why there is potential research in FA and also in other genetic neurological diseases that are considering direct injections into the brain. Lipid nanoparticles and extracellular vesicles, which are being researched because of the issue with the immune response, which can sometimes prevent people from having multiple genetic therapies. There's ongoing research with them, and we know lipid nanoparticles right now mostly go to the liver. So there's research ongoing to see if they can be redirected to the organs or tissues of interest.
Well, and another question, so I know that for ex-vivo, you're taking someone's cells out of their body and then giving those cells the genetic therapy and then putting them back in. So what actually happens when the cells that have received the genetic therapy are put back in the body?
Thanks, Chandra. I'm going to answer that one. Do we want to go back to that slide? Can we go backwards and just look at that slide? So there is a potential therapy in the pipeline that you'll see that's been researched at an academic lab, and a company is now interested in bringing that forward for an ex vivo approach. In that case, the cells that are collected and that are going to receive the genetic therapy are hematopoietic stem cells. So the stem cell part is an important part because that's a cell that can give rise to other kinds of cells. Those hematopoietic stem cells are removed from the bone marrow or from the blood. And then in the case of the one program in the pipeline, they're genetically edited to take out the GA repeats, cultured. And what has to happen to people that would receive this is it's very similar to an oncology approach. You have to have a bone marrow transplant. In that case, you're taking some kind of chemotherapy to eliminate your own endogenous cells, and then these engineered cells would be transplanted back into your bone marrow. Because they're stem cells, they can migrate to other parts of the body, including the heart and even the brain, and they will become a certain kind of immune cell. that in the presence of damaged tissue, they can help to support that damaged tissue. So that's our hope for the one ex vivo program that's currently in the pipeline.
Thank you. I just have one more question. So you mentioned a couple of different rare diseases that have approved genetic therapies, but FA is not one of them. So are we behind?
So like I mentioned before, one of the major differences between FA and the rare diseases that do have approvals is FA has multiple different organs and tissue types that need to be targeted in comparison to many of the diseases that do have approvals. So for example, another one on this slide, Luxturna, Leber's congenital amaurosis, that is a kind of blindness that occurs very, very early on, usually at birth. So for that therapy, only the eyes had to be targeted. The fact that other rare diseases do have these approvals, and every one of these approvals behind them are several years of clinical trials that investigated the safety and efficacy of these genetic therapies and of the viral vectors that were used for some of these therapies. That data helps us understand how genetic therapies can be used for FA, and that is across the rare disease space. The more we learn about genetic therapies in general in any rare disease, the more information we have to use for treatment specific for FA or for other rare diseases.
Okay, thank you.
Thanks, Chandra.
Well, Kellen, Chandra, thank you for taking us through an introduction and quickly into the very specific details that we want to get to related to developing genetic therapies for FA. Maybe just a quick overview before I go to the FA specific topics. What I hope you took away from the first part of our discussion is that there are a lot of reasons to be excited about genetic therapies. There are advantages to them. You are trying to treat the disease at its root cause. And hopefully if we, only have to do these therapies one time, they won't require repeat administration. They will hopefully be able to be combined with other therapies like the small molecules, or maybe other genetic therapies with other delivery approaches in the future. But there are risks and challenges associated with these genetic therapies. And these are not unique to FA. These exist for all genetic therapy approaches. But the big one that you heard Kellen mention and start to talk about is related to the fact that, viruses are currently the main vector used to deliver. And when you are exposed to a virus, your body recognizes it as foreign and mounts an immune response. And we've learned from some of the earlier gene therapy trials that came before the FA trials that if you give high doses of viral vector, it will cause immune response and serious adverse events. And so we want to be careful in terms of, the therapies as we're developing them. We want to be able to reach all these target tissues, but we can't do that by simply upping the dose. And that's why we're thinking about multiple routes of administration. So just hopefully this is helpful recap of kind of some of the advantages and disadvantages for the field. So now when we think about genetic approaches for FA, what are some of the advantages and challenges that we have that are FA specific? So many of you hopefully know kind of the basics of Friedreich's ataxia genetics. FA is caused by mutations in a single gene called Frataxin. And the good thing is Frataxin is actually a small gene. And so that gene can actually fit into lots of different types of delivery vehicles. That's not the case for all genetic conditions. Some genes are quite large and can't fit into the delivery vehicles. So we have a small gene. And as Kellen told you, most people with FA have the same mutation this GAA repeat expansion. And that fact that most people have the same genetic mutation means that gene editing approaches are also, very advantageous for FA. You don't have to try and make or have thousands of different genetic editing approaches. Some diseases, every single person who has the condition, they have a different mutation in their gene. And so gene editing is really challenging in those cases because you need hundreds, thousands of different editing versions. For us, it's the same mutation we're trying to edit in most people. So both gene replacement and gene editing are viable approaches in the context of FA for some of the underlying genetic basis. We've also been able to do some of the proof of concept behind gene replacement and gene editing in the FA cell and animal models, and have been able to demonstrate kind of this proof of concept of both of them. Another important aspect when developing genetic therapies for FA is, the gene mutation, as Kellen talked about, causes a decrease in this protein, frataxin, that's an essential protein in the body. And FA is often thought of as being caused by this frataxin deficiency. However, individuals with FA do have some frataxin. They're not completely null or void of the frataxin protein. And this is important in the context of genetic therapies because some genetic conditions where you don't make the protein at all, when you administer the protein in a gene, in the context of gene replacement, the body recognizes that new protein as foreign and can mount an immune response to the therapy you're trying to give to the new protein. In our case, this is unlikely because people with FA make some frataxin, they just don't make enough. The other advantage we have is that the mutation causes a deficiency in frataxin, not an abnormal version of frataxin. And so we don't have to get rid of an abnormal protein being produced. Finally, another advantage we have is that we know that carriers, parents of children who have FA, don't have symptoms of FA, yet they don't make 100% of normal frataxin, they make about 50%. And so we believe that increasing frataxin, even to modest levels, could have a therapeutic benefit. And so all of these are advantages to trying to pursue genetic approaches for FA. And again, treating the disease at its root cause makes a lot of sense. I think I remember the very first questions I got from people in the FA community. And everybody always asked me, even 25 years ago, when I started out, why can't we do gene therapy? Why can't you just replace the gene? Or why can't you just replace the protein? So these kinds of treatments, I think, make a lot of common sense to us. And the fact that they can have a durable effect is also exciting. Helen also mentioned, the 37 trillion cells that make up our body. We know through our natural history studies a lot of the symptoms that are caused by FA and how they progress. And we know that FA affects several different areas of the nervous system, like the cerebellum, like the spinal cord. It also affects other tissues outside the cerebellum, like the heart and the pancreas and the skeletal muscle. And it's good that we know these targets for therapy, but it's also challenging in the context of delivering the single genetic therapy to all of these multiple targets simultaneously in one gene therapy. And I mentioned this at the beginning, that we can't just try and solve this problem by increasing the dose. And we know that both because we're limited in terms of how much viral load people can be exposed to, but also some of the work done early in the FA mouse models showed us that if you increase the dose too much, you can also get frataxin toxicity. And this was a surprising finding for the FA community when we first learned about it. But I think it makes sense when you really think about it. So our challenge with FA is that if you have too little frataxin, what this slide is trying to show you, maybe I should back up a second, is what happens to cardiomyocytes or heart cells when there's too little frataxin. So on the left, cardiomyocytes don't have enough frataxin, and that leads to cell death. On the right are cells that have been given 20 times the normal amount of frataxin. Again, 20 times higher than the normal level, so really high levels, but they also result in the same thing, cell death. And so what we've learned is that there's this thing called a therapeutic window, and we have to be in that therapeutic window. We want to increase frataxin, but we can't increase it too much. The good news is this therapeutic window is pretty wide. It's not narrow. It's not like threading a needle, but it also means we can't solve all of our problems by just upping the dose. So just to sum up, there are advantages for genetic therapies for FA, and there are challenges. One of the challenges I didn't cover yet, but I just want to bring up, is that ideally when you're starting to develop a new medicine of any kind, after you administer it, you want to know, did it get where it was supposed to go? Is it where it's supposed to go? Is it doing the thing it's supposed to do? And this is really challenging for us when we think about delivering genetic therapies to the brain and spinal cord, because we don't have that window into the brain or the spinal cord that can tell us in a very short period of time after dosing, did the genetic therapy get there? How many cells did it get to? Is it doing what it's supposed to do? And because these therapies are permanent and irreversible, that does give us a little bit of pause that, we want to be really careful before we go from animals to people, that we really have the right dose. And if we had biomarkers or tools that told us, that helped us answer these questions, that would be something that would be a big help. And that's an area of research we're actively trying to work on, is to find these kinds of biomarkers into the brain and the spinal cord specifically, because that would really help accelerate these treatments for the brain and spinal cord. And so I'll stop there and take some questions.
Hi, Jen. Thanks so much for that. Just a couple questions for you, and I'm sure there's much more, but to start with a couple questions. Does the age someone take the therapy matter? So is it better to get it when you're younger or older? What about children? Would they benefit from taking it?
It's a great question, Becca. And the short answer is we don't know. And that's why we need clinical trials, right? Some of the learnings we have from some of the other diseases that have had clinical trials of some of these therapies have shown that, the earlier you intervene, the earlier you administer the therapy, the larger potential benefit individuals have received. And that makes sense because we can't rescue cells that have died or have degenerated. We're trying to rescue cells that are not functioning properly. So, we do believe that the earlier we intervene, hopefully the bigger potential benefit individuals will have. We do have evidence from the FA mouse models that not only can these therapies help prevent symptoms, but we also do see recovery when the animals are symptomatic. So we do believe that there's the potential, even for people who've had the condition for a while, to have benefit from these therapies. And maybe the other challenge we have, FA takes place over a long period of time, right? Decades, and the symptoms evolve and change. And so the therapy that might be best suited to somebody newly diagnosed might be different than the therapy that's best for somebody who's had the disease for 10 or 15 years in terms of what target tissues, what symptoms you're trying to treat. So hopefully that answered your question, but it's a good one and it's complicated.
Thank you. Yeah, so I think I heard you say that even it's if we can't, to take it earlier, but even like adults who have advanced disease would still benefit as well.
I think potentially depending on what symptoms, right? People who have had FA for quite a while start to have vision loss. And so trying to target the vision loss of FA could be really valuable for people who have had the disease for a longer period of time, or trying to treat the the cardiomyopathy becomes very aggressive in adulthood. And so, I think it depends on the symptoms we're trying to target in terms of when we're targeting them.
Perfect. And then a follow-up question. Are these genetic therapies a cure for FA, or would we still need other treatments like Skyclarys or heart medicine or whatever we need to
Yeah, I'm so
kind of
glad you
function?
asked that question. A lot of times I think these therapies are really exciting. And in the lay literature, a lot of times we see them put forward as potential cures. And we all want a cure. That's why FARA's URL is Cure FA. But these are not cures. These really are treatments. And it is likely that we are going to need several treatments combined to achieve our goal of slowing, stopping, reversing FA. And even the genetic therapies are going to have to be used in combination with other therapies.
Thank you, Jen. And then are there any general risks for someone with FA to take these genetic therapies?
There are risks with every therapy. I think that's important for everyone to keep in mind. And it's important to always understand what those risks are and talk to your doctor or the clinical study team if you're in a trial about what the potential risks are. We talked about some of them, I think, today. There's the risk of an immune response in response to a potential viral delivery. There's risk for frataxin overexpression, for example. And in the context of gene editing, we haven't quite covered this yet, but some of the gene editing technologies are more specific than others, but there can be off-target effects. When you think about editing the genome, how do we know we're only editing that one spot that's important to us, and we haven't done anything to all the other 30,000 genes?
Thank you so much.
Thanks, Becca. Maybe just a quick update in terms of where we are in the development of these genetic therapies for FA. So this is the bottom part of the pipeline, blown up a little bigger so we can all look at it together. And we've had significant progress in developing genetic therapies for FA. We have 3 in clinical trials right now. They are all trying to treat the cardiomyopathy of FA. So Lexeo Therapeutics and Weill Cornell, they have very similar programs. It's the same genetic therapy. It's administered through an IV administration, and it's trying to treat the cardiomyopathy. Astellas Therapeutics announced recently that they also have a gene replacement approach, again, to treat the cardiomyopathy of FA. In the next few weeks, we're going to have webinars, one from Astellas and another one from Lexeo, where they're going to be able to take you all through a much more deep understanding of their specific approach, their genetic therapy approach, and their trials and kind of where they are in development and get into a lot more detail than what we can cover today. But we do have genetic therapies now in clinical trials. That's an important milestone for our community, I think. Other therapies are earlier in development. And again, just a reminder, if we're looking at the left side of the slide, things that are in discovery, are still very early in development. In the middle, you're kind of doing what we call preclinical studies, doing all of the manufacturing, large animal studies, getting regulatory approvals to be able to begin clinical trials. We've got programs that are trying to target the retina or the eye, both gene replacement and gene editing. And we do have other groups that are working to try and treat multiple targets at the same time. So the brain, spinal cord, and potentially the heart. And they're doing those in slightly different ways. One of the programs, the FA212 program, is one where we would be doing multiple administrations at one time. So direct intraparenchymal into the brain delivery to treat the nervous system, as well as an IV to try and treat the heart. Voyager Therapeutics and Neurocrine, they've been working on developing the next generation of viral vectors. And they're hopeful that they'll have more efficient vectors that will be able to deliver to more target tissues with a single administration. Again, these are earlier stage. They're not in the clinic yet, but I hope you see there's lots of work going on to try and solve some of the challenges, and there are going to be different ways, different programs are going to try and tackle those challenges. There are gene editing programs, both in vivo, so Prime Medicines, for example, they have a genetic editing approach that they're hopeful will treat multiple organ systems. And then there is an ex-vivo program that Barbara talked about earlier. So maybe I'll stop there. Take a few questions.
So I saw that there are like 2 different gene therapies for the heart, and there's 2 different ones for multiple tissue approaches. So how do we tell the difference when there's more than one?
Yeah, great question, Chandra. Thank you for that. I think it's important for everyone to realize you should ask lots of questions. And there are lots of ways in which these genetic therapies can be different from each other, even if they sound the same or look the same on the surface. So the delivery vehicle could be different. Even though it's an AAV, what this slide is trying to show you is there are many different types of AAVs. And those different AAVs have different affinity for different target tissues. And so while the 2 programs that are in clinical trials might both be targeting the heart, they could have different delivery vehicles. They could be using different types of AAVs. It's also important to know what goes in the vehicle, right? What's inside? We made it sound really simple. Oh, we're just going to put the gene in. But it turns out that, yes, you're putting in the gene, but you also have to add other regulatory elements so that when the gene gets into the cell, it actually gets expressed. It makes the RNA and protein. And that can also make one genetic therapy a little bit different than the other in terms of, what promoters or on switches it has. Is that specific to a certain cell type, or does it express in all cells? There's some programs now looking at being able to develop off switches, because we talked earlier about how these are permanent changes. And there might be, and we talked about Frataxin toxicity being a potential issue. We might want to be able to turn off a genetic therapy. And so there are new technologies being developed where maybe an off switch can be inserted into what gets delivered. And related to gene editing, Helen alluded to this, that there are different types of gene editors, just like, so there are all different kinds of, if you use our little analogy of the scissors, there's CRISPR, there's prime, there's base, there's zinc fingers. There are all these different kinds of editors that have recently been discovered. And those different editors are going to have different efficiencies for the types of genetic edits they're trying to make. And again, I mentioned earlier that there could be off-target effects of gene editing. And so really understanding what the locators are being delivered so that it only edits where you want it to edit. And so those are some of the kinds of things you can begin to ask about when you're considering or trying to learn about one genetic therapy compared to another. Is that helpful?
It is, thank you. Kind of following off of that, so it's very exciting that there are a lot of genetic therapies in the pipeline. How do I decide which one I should do?
Again, these are great questions. We could probably spend an hour just on this one. But it's really important to learn as much as you can before you participate in a clinical trial. And, you know, asking some of the questions like the ones I just, you know, shared with you, as well as, you know, learning as much as you can about what it means to be involved in the clinical trial. What phase is it? What's already known about the drug? What, you know, what preclinical, what animal studies were done? Have there been clinical studies done? What do we know from other clinical trials? These are all questions that you should be asking the physicians and the study team where they're at the sites where they're conducting clinical trials. Also, about a year ago, FARA released what we called a clinical trial guide. And in that clinical trial guide at the back, there's a whole bunch of questions that I hope are useful to you, to the community, in thinking about what questions you can ask. But we put together a few more questions that are specific for genetic therapies. Some of these are questions that help you evaluate the safety and efficacy. So they go over some of the things we've already talked about, but some of them are questions that hopefully help you think about what's important to me. What symptoms do I want treated? Am I able to participate in this clinical trial? Will this impact my ability to participate in other trials? And how do I feel about that based on where I am in my FA journey? And so we're going to be sharing these this update to the Clinical Trial 101 guide and making that available on the website for everyone.
Thank you, and I just have one last one that's a two-part question. So first of all, could I take a genetic therapy in addition to something else like that's already approved, like Skyclarys? And then the second part is, could I take multiple genetic therapies? For example, one that's targeting the heart and then one that's targeting the nervous system later?
So when therapies are approved, you're likely able to combine them. And certainly when something's like a small molecule like Skyclarys and a genetic therapy, it's likely that those can be combined. Where we run into challenges is if a genetic therapy is using the same virus. For one genetic therapy uses virus A, the next genetic therapy also uses virus A, then you can't take both, because your body will have mounted an immune response to virus A after the first time you see it. And so in some cases, yes, we're going to be able to see combination therapies, and in some cases, no. I know, for example, with the Lexeo gene therapy trial, they are allowing people to be on Skyclarys. It's an approved medication. And you can be on Skyclarys and you can still participate in that clinical trial. But these are all important questions for you to think about and contemplate, both for clinical trials as well as for managing your FA. Barbara, I'm going to turn it over to you maybe, and if you can talk a little bit about the other things FARA is doing to try and help develop genetic therapies.
Absolutely. Thanks,
Before we do, I'm sorry, I didn't, the second part of that question, could you take multiple genetic therapies? Like, could I do one for the heart and later do one for the nervous system?
Possibly yes and possibly no. The possibly yes is if the one genetic therapy for the heart uses, let's say, a viral vector A, and the next genetic therapy for the nervous system uses a completely different viral vector or a non-viral delivery, then maybe those can be combined. But if they're both using the same delivery vehicle, it's unlikely that you'd be able to take both because it would be less effective, more chance for side effects.
Okay, thank you. I just wanted to clarify that.
Yep. Thanks, Chandra.
Thanks. So FARA has been doing a lot, but none of this has been done without the community's support. And, you know, we're very grateful to the community, both for the fundraising support that we get, but also for the participation in trials. As Jen said, we've learned what we've learned because people have been heroes and stepped forward to participate in genetic therapy trials for other indications. But it's really also the underpinning of everything that this community has supported the natural history studies that we've done. That gives potential developers an understanding of the disease, and they need that in order to commit to developing a therapeutic. So in addition to that the community has done, FARA has funded science. You know that we have a grant program. We funded some of the work that Jen actually cited earlier about showing in animal models that it is possible to do gene replacement and that you can rescue, therefore, the animal model that is modeling FA with a gene replacement. And, you know, supporting companies as well to develop that proof of concept in the model systems is really important for those companies to be able to go forward. More recently, we've been really trying to bring other scientists that are outside of the FA field into the FA field. As Jen was saying, some of this work has been done in other indications, and that learning, we want it to be applied to FA. So we've been encouraging scientists that are developing these very novel approaches, like prime editing, to come in and try that in FA. And finally, the novel delivery mechanisms are sort of the next frontier. The frustration of the being able to treat multiple target organs with a single virus, that is a problem that needs to be solved. And that's a place that we're spending some time. In the clinical realm, Jen also talked about the importance of biomarkers, and especially for the nervous system, it's a black box for us. We can't look at Frataxin in the CSF, for instance. That doesn't give us really actionable information. So TRACK-FA, the imaging study that's been underway, which many of you have volunteered to be part of, is the first really good longitudinal study for us to look at the brain and the spinal cord changes over time. And that's going to be fundamentally important for programs that go forward to treat the neurological symptoms. And of course, the clinical designs are all based on those natural history studies. And FARA does a lot to help developers think about the right patient population, the right endpoints to use. MFARS was developed from all of your efforts, and that's an endpoint that now we can use in clinical trials. In addition, gene therapies are different from small molecules in that they require an infrastructure that's a little more sophisticated, a little more complex. The logistics are a little more complex, and FARA is spending time internationally talking to sites that could be sites for clinical trials in genetic medicines, in genetic therapy, to help them be ready, not just the clinician that's going to develop or administer the genetic therapies, but the pharmacy, even the institutional review boards need support to learn more about these really cutting edge therapies. In addition, some of you will remember a couple of years ago, FARA knew that it was a frustration of some of our sponsors that are trying to move forward these really novel medications that they couldn't get the attention of the FDA in particular. And we were able to bring together a couple dozen sponsors, as well as a couple dozen people at the FDA, to talk about the challenges of genetic medicines, specifically for FA, so that we had some baseline alignment on what can be done, what's still very challenging, and where we need to make some progress. In addition, many of you here have helped the FDA understand, your interest and your weighing of risks and benefits when it comes to these kind of therapies, and that's been hugely helpful to regulatory agencies. This is the year of pediatrics. We are really interested in understanding how early we can go, but some of that requires that we develop some other mechanisms of measuring progression of FA in children, and that's another frontier that FARA has been working on. And FARA is always there when industry partners go before regulatory agencies, either inside or outside of the US. FARA is always there to attend and to help both the agency and the sponsor understand the community, understand what's important, understand how to help the community get the kind of therapeutics that they deserve. And finally, helping you understand what these medicines can and cannot do, what they require of you, and what we still don't understand and are still learning. I think that education piece is always going to be part of FARA. And I'm going to stop now so that you can ask a couple more questions, because it's really important to us that you have the information you need.
Thank you, Barbara. Becca, I know you've been watching the chat and the Q&A box, and I know you still had some more questions yourself, but maybe since we're close to the top of the hour, I really like for us to get to some of the questions coming in from the people joining online. So if you see some of those questions that you want to bring forward, please do.
Thank you so much. Yeah, one of them that I've seen a couple times is, will FARA support the development of gene therapy trials outside of the US? So if somebody lives in Spain or India, will they have access to these gene therapy trials?
Yeah, that's a great question, Becca. And you know that this is also the year of international support and development of our international footprint for FARA. So the combination of the natural history study that was traditionally done in Canada, Australia, and the US, with the combination of the natural history study that was done in Europe to make one unified study now, has helped us to really align across the world on how we would do these clinical trials. And there's also been an effort to make sure there are sites outside of the US so that the clinical trials can actually be run in many different locations. So the FA Global Clinical Consortium, or GCC, is an effort to make sure that there are sites in other geographic areas so that eventually everybody would have access.
And I'll just add that, a lot of our development partners are looking outside the US. They want to be able to conduct trials in multiple countries simultaneously, and they are actively working towards that.
Thank you. I had a couple questions about mouse models. Has Ex-vivo been tested with FA mouse models? And if so, which model type, what tissue types as well?
Ex-vivo has been tested. Jen, the program that Jen showed you, the UCSD program, that's also now being partnered with a company called Papillon, is through a FARA-funded investigator, Stephanie Cherky, who started looking at ex vivo gene replacement therapy for other rare indications. and actually did show in the animal models that gene replacement therapy through ex vivo can show a benefit in some of the FA models. She's now moved on to looking at editing, and FARA has helped to facilitate that and continues to support her to move that project forward.
And we're also supporting a few other investigators in the community to look at the same approach, but in different animal models. So can we see benefits in the cardiac model? Can we see benefits in neurologic animal models? So we're trying to build to really understand by testing multiple different animal systems, not just one.
Thank you guys so much. And then I had one question. We talked, or Barbara, I heard you say that there was a year of like pediatrics and a year of international. Is there a specific approach or specific projects that FARA prioritizes, and how does FARA decide what to prioritize?
Yeah, those are great questions. You know, there's levels of what FARA does. So at the science level and the very basic research and our funding, we have prioritized to try to push on the frontier of getting more, I guess, less delivery mechanisms that would have the immune liability. That's the next frontier. It's it's still very, very early, but there are possibilities of using non-viral approaches, and FARA has helped to fund some of those, both in academic settings and actually in a company as well. We're also very interested in moving the gene editing forward. And we talked about prime medicine. That program actually came out of David Liu's lab at MIT, and he's one of the investigators that's funded through the Friedreich's Ataxia Accelerator program at MIT and Harvard. I'm trying to think what else. The ex vivo work as well, as Jen said, to really try to understand that, make sure that we get to the mechanism so that we have a clear, it's not a black box, we have a clear view of both the possibility of efficacy, but also if there's any risk associated with that. And then at a higher level, you know, those engagements with international sites to make sure that they are well positioned to be able to run clinical trials. And as Jen said, that's an easy sell to sponsors. They're excited about the opportunity to do things outside the US.
Thank you so much. And I saw one question pop up a couple times, so I wanted to make sure I pose that to you as well. Can embryonic stem cells be used, or things like cords, like spine, or cord storage and blood storage? Would those be potentially beneficial for treatment?
So embryonic stem cells are a pretty old technology, and we've really moved beyond that. In science, it's not necessary to use embryonic stem cells now. Some of the model systems that we use are from the fibroblasts that you all donated, and they can be de-differentiated, so they can be moved back to an earlier stage, and then they can be used to become a much more mature cell model and potentially for a therapeutic as well. For cord blood, really what you're getting out of cord blood that could be useful is those hematopoietic stem cells. So that is a source, but there are other sources that are probably a lot more, efficient to get to, including just a bone marrow biopsy.
Thank you so much.
Well, I know we're a few minutes over the hour, and I'm glad we were able to get to some of your questions. It's been exciting. We've seen, at one point I saw over 145 people in the webinar, which is terrific. Hopefully we got to a good number of your questions. Again, there are going to be 2 more webinars coming up in September. One, Astellas is going to talk about their genetic therapy program in FA. And then Lexio later in the month on the 24th is going to give an update on their gene replacement approach. And then we also have our symposium on October 7th in King of Prussia. That's going to be both in person, and we're also going to stream it. So lots more opportunities to continue this conversation, continue our learning and our discussion. And so with that, I would like to thank all of you for attending and participating today, and especially thank Becca and Chandra for joining and helping moderate our discussion today, along with my colleagues, Barbara and Kellen. Thanks, everyone.